A very big hello to all my wonderful KCET champions out there. I'm so sorry for the break that we had in between. I had some medical emergency in my family, but now it's fine. And now I hope we are also back on track. Well, I'm very energetic today because we are going to deal with KCET PYQs. So my dear children, are you ready for scoring 60 on 60? Well, I hope you're ready. And if at all you're ready, definitely let me know in the comment section below. And for those of you thinking that why is this ma'am very energetic today? Or why is she overreacting? Well, there's a reason for it, because I have put in my lots of efforts into structuring this video so well that you understand each and everything of this particular chapter. It is going to be really interesting for you guys because this is where you can participate really well. There will be questions, you can answer me in the comment sections. Yes? So my dear children, for those of you who do not know, we have completed this chapter in 3 sessions. Okay, it is there in the YouTube channel. Please watch it. If at all you feel it's lengthy, you can watch it in 2x speed, but you have to give it a watch. Now we are going ahead to test the knowledge by solving the previous year question, and I have segregated this video into 4 parts. There will be round 1, which will be 2025 edition, as in the questions that came this particular year from this particular chapter, that will be put down first. Let's see if you're able to solve that first. Yeah, and then there will be round 2, which will be numerical based questions, something that bio students usually do not like, right? So that will be there. The previous year questions that came, which was numerical based, it will be solved in round 2. Now round 3 will be theory based question. If you know the theory, you will be able to answer it really well. Now round 4 will be diagram based question. So I have taken questions from every nook and corner of this particular chapter, and I'm going to make you solve that today. And with this, you will get that confidence that yes, I have completed sexual reproduction flowering plants completely, and I'm gonna get 11 marks in my PU2, and in KCET I am going to grab those 3 marks, and those 3 marks are really important. So I welcome you to the Sankalp session that we have today. And my dear children, if you want to download the notes, it is there in the WhatsApp channel. Please be a part of our WhatsApp community. And if at all you feel that this particular video is going to help you really well, do not forget to hit the like button right now, and also do not forget to hit the subscribe button for getting all the updates regarding the next chapter that will be coming in. So before I go ahead, my dear children, we have brought in so many rank holders the previous year, and I want you to take this position the next year, and for that you have to work really hard. Yes, and I'm pretty sure you're gonna do that. And also one more thing, my homework winners are also here. So I see a lot of people are now participating. Okay, they are giving me the answers. I'm really proud of you. Big kudos to you guys. We have Deepika Manohar, we have Putta Raju Karthik, and we have Manvita Reddy. We have Abhi 0007. All of them gave me the correct answers, and big kudos to you. You were correct. Homework 1 answer is meiosis, and homework 2 answer is epiblast. And I want each of you to participate as well. It is really important. So kudos to all of you. Let's go ahead. Let's hope that this time we have more winners out there. Now, instruction. There are certain instructions before. Okay, do not be worried. Yeah, instructions before we go ahead with the KCET PYQs. Let's see what are those. So you have to read the question carefully and attempt by yourselves first. So whenever I show you a question, pause the video, attempt by yourself, give me the answer in the comment section. That is how it should work, in the comment section. Okay, the question number 1, like that you have to tell me. Now in case you get wrong answer, note down the question number and attempt it again towards the end. Once you have finished the whole video, you can re-attempt the question, because that is how you will understand from your mistakes. Okay, so that is there. Now no cheating. Please go through all the lecture videos. If needed, use 2x speed. After watching those videos, I'm pretty sure you will be able to answer each and every question that is going to be shown up over here. Okay, so are we ready? Hope you're ready. Let's get ahead with round 1, the 2000, not 24, it's 25 edition. The first question that we have that came this year: when the pollen grains of a flower of a plant pollinate the stigma of a flower of another plant, it is called. So they have given a scenario. There are 2 plants. Okay, if the pollen grains of a flower, that means there is a flower over here. That's the anther. Now the pollen grains over here is going to pollinate the stigma of another flower. Stigma of another flower, so we have to make that over here. Okay, it is called as what? So from here the pollen grains are going, this is going up there on the another plant. This is plant 1, and this is plant 2. What do you call this type of pollination? Do you call it dichogamy? Do you call it geitonogamy? What do you mean by geitonogamy? Geitonogamy is when in the same plant you have two flowers. Suppose this is the male flowers having the male anther and everything, and there will be female flowers as well. From here to here if it goes, this is known as geitonogamy. That happens in the same plant but two different flowers. Now xenogamy, what do you mean by that? Xenogamy is where you would be having cross pollination. What do you mean by that? That is, there will be two plants involved over here. From one plant, the pollen grains are going to land on the stigma of another plant. It looks like an X over here, right? So you can remember xenogamy is for cross pollination, correct cross pollination. Well, when this happens, this is also cross pollination. This is also cross pollination. But my dear children, will you be able to see any genetic variation? No, because the flowers are belonging to the same plant. But in the case of xenogamy, where two plants are involved, you will be seeing genetic variations as well. Now, autogamy is the next option that we have. Autogamy means basically you need to have both the male and the female part over here within the same flower. If pollination is happening, that is known as autogamy. But here, those are not the scenarios. The scenario is the pollen grains of a flower of a plant is going to the stigma of another plant. Okay, please understand that. So here cross pollination is happening properly, true cross pollination is happening, and this is known as xenogamy. If at all it was happening from here to here, still it is cross pollination, but it is not true cross pollination, or we say genetic variations would not be there. It is geitonogamy. Okay, and if at all it is happening within the flower itself, it is known as autogamy. So your answer would be xenogamy. So let's see who all got it correct. Question number 1, the answer is xenogamy. Now, the next question that we have is fusion of a male gamete with the central cell in the embryo sac of an angiosperm is called as. What do you call that as? So my dear children, if you see the embryo sac structure, you have 3 cells at the top. These are known as antipodal cells. You have 2 cells over here. These are known as synergids. And you have 1 cell over here, which is the egg cell, the female gamete. Right. Now over here comes 2 male gametes to fuse. So the 1 male gamete that comes initially is something that would be fusing. It's not visible, so I'll make it visible. Yeah, it will come and fuse with the egg cell out there. This is 1 male gamete, and the next male gamete which is there, it is going to fuse with something else right over here. There are 2 polar nuclei, and this whole structure that you see, my dear children, is a big cell. So the big cell with 2 polar nuclei, it is known as the central cell. So I can say the next male gamete that comes in, there are 2 male gametes coming in, the next male gametes that comes in will be fusing off with the 2 polar nuclei of this central cell. And what do you call this? What do you call this? Do you call this syngamy? No. Syngamy is when the male gamete and the female gamete fuses together. This is known as syngamy. Do you call it apomixis? No. Do you call it double fertilization? No. Do you call it triple fusion? Yes. Why? Because the male gamete has 1 nucleus, right? Here you have 2 nucleus. Okay, the central cell has 2 nucleus. So 1, 2, 3. Since 3 nuclei are fusing, we will call this as triple fusion. So your answer would be this. Fusion of a male gamete with the central cell in the embryo sac of an angiosperm is known as triple fusion. Remember, this fusion is triple fusion. This fusion is triple fusion, and this fusion which is there between egg cell and the male gamete, it is known as syngamy. So since 2 times fusion is happening, you call this as double fertilization. You call this as double fertilization, but that is not something they have asked. They have specifically asked about central cell and the male gamete, so it will be triple fusion. Why? Because 3 nuclei are coming together. I hope that's clear. So what is your answer for this question? It is D, triple fusion. Now the third question which have come this year is, which of these option is true in the context of the above diagram of pollen grain? So they have given a diagram of pollen grain, and over here you can clearly see there is a big cell with a nucleus and there's a small cell with a nucleus. So the big cell which is there, you should first identify it before going ahead reading the whole thing. So the big cell, always it will be the vegetative cell. and the small cell always it will be the generative cell. Okay, now you can read the other option. A is a generative cell. Is A the generative cell, my dear children? Wrong. So do not waste your time. Wrong. Okay. A is vegetative cell. Okay, A is vegetative cell. Okay. With abundant food reserve. Yes, it is very big because it has abundant food reserve, because it has to nourish what? The B. That is the generative cell. Why does it have to nourish the B, generative cell? Because generative cell is something that forms the male gametes. The generative cell will be dividing itself to form 2 male gametes out there. This is the pollen grain where all of this thing is happening, my dear children. So vegetative cell provides nourishment to the generative cell so that it can divide to what? 2 male gametes out there. So don't you think that this would be your answer? Before going ahead reading with any option out there, you got your answer, but you still have to go ahead. Okay. Now A is generative cell. Wrong. A is a vegetative cell. Correct. Which gives rise to male gametes. Do you think this is the one that gives rise to male gametes? No, it is for nourishment. So again, this would be wrong. So what is the correct answer? It will be B. So with this, we have got 3 marks in KCET this particular year itself. You have solved those 3 questions which came in the year 2025, my dear children. I hope you got it. And if at all you were able to solve it by yourself, do let me know in the comment section. My dear children, remember you have to tell me your whole score towards the end of the session, okay, in the comment section. That is only how I'll get to know whether you are able to understand things or not, and I'll be really happy to see your actual scores, okay. Now going ahead with round 2, numerical based question. Now that is going to tickle something in your brain out there. The first question that we have over here: a flower has 10 stamens. How many stamens? 10 stamen. What is stamen? Stamen is the male reproductive part made up of anther and filament, right? Now 10 stamens each having bilobed dithecous anther. So just to confuse you guys, you just understand that basically all the stamen will be having bilobed anther. Fine. Now if each microsporangium has 5 pollen mother cell, how many pollen grains would be produced by the flower? So for that, you should have an understanding of the structure of the anther first. So my dear children, when you say one stamen, this one stamen has an anther that looks like this. If I cut it and I see it, it looks like this. It is bilobed, as in it is one lobe, and this is the second lobe. Each lobe that you see over here, you have 1 microsporangium, 2nd microsporangium, 3rd microsporangium, 4th microsporangium. How many microsporangium? 4 microsporangium in one stamen. Now inside the microsporangium you have what? The pollen mother cell. Now over here it is said that each microsporangium has what? 5 pollen mother cell. So can I write 5 over here? Agreed. Now each mother cell, if you take one out of it, okay, if you say there are 5, 1, 2, 3, 4, 5, for your understanding I am doing that, okay, 5 pollen mother cell are there. Each of them are going to undergo meiosis, okay, each of them are going to undergo meiosis, and at the meiosis, my dear children, at the end of meiosis, how many cells do you get? You get 4 pollen cells, okay, or basically you get 4 pollen grains at the end from 1. And how many of them are there? 5 of them are there. So 5 into 4, you will get what? 20. Don't you think that you will get 20 pollen grain cell? How? Because each of them are going to give rise to 4 pollen grains. So 5 of them are there. So 5 into 4, you get 20. So 20 over here, 20 over here, 20 over here, and 20 over here. So can I just do 20 into 4? 20 into 4 is what, my dear children? 80. Is my answer complete? No. How many stamen are there? 10 stamen are there. So I have to do into 10. So your answer would be what? 800. Okay, so your answer would be D, 800. Always have that understanding. One stamen, okay, would be having 4 microsporangia. Okay, and each microsporangia will be having indefinite number of pollen mother cell. Suppose here they have given 5, so you have to take 5. Now each 5 of them would be giving rise to 4 pollen grains each, so we will say 20. Now how many microsporangia are there? 4 like that. So you do 20 into 4, you get 80. Now how many stamens are there? 10. So your answer would be 800. I hope it's clear. Okay. Always remember that this part of the question is just to make you confused. That's all. Okay. Now I hope you got it. Not forget to hit the like button if you're understanding things, okay, and do let me know in the comment section. Now we have the next question, numerical based question. Yeah. in apple, the chromosome number in the gametes is, okay, in the gametes is 17. Okay, 17 in the gametes. Now, what is the chromosome number in its primary endosperm nucleus? Any idea? So, my dear children, we have to form the embryo sac again. It's about the gametes and the PEN. So, embryo sac, the female embryo sac has 3 cells out there. What do you call them? Antipodal cells. Okay. 2 cells over here, you call it synergids. You call them synergids. One more thing I want to explain you here itself. When you talk about the nucleus of the embryo sac, now all of the cells have haploid number of chromosomes. That is half the number of chromosomes. For example, a plant has 24 number of chromosomes in its normal cell. Then in the cells of the embryo sac, it will be half of it. 24 ka half? 12. It will be 12, 12, 12, 12, 12 over here. So these are the antipodals that we have. These are the synergids that we have. What else do we have? We have to have the female gamete, right? So the female gamete is also there again with how much number of chromosome? Half number of chromosome. So this female gamete which is there, the egg cell which is there, it has half the number of chromosome. Okay, now you have one more cell. I told you previously also, what is that one more cell that you have? It is the central cell. Now the central cell which is there, it has 2 nuclei. It would be having 2 nuclei right over here. And each nuclei, my dear children, all of the cells has nucleus, has n number of chromosomes, right? So this particular cell has 2 nuclei, so it becomes 2n. It will become 2n. The cells chromosome number, if you see, it will be 2n. Now, my dear children, the first male gamete which is there, it comes and fuses with this. So the male gamete which is there, it will also have n number of chromosome, half number of chromosome. N and N comes, you get the zygote with 2N. Okay, now it will be just like a normal plant cell. So everyone were 24, they were 12. So 12 and 12 came together. Now in the zygote, it is 24 again because 2N has reached. Now over here, it is already 2N. Again, one male gamete is going to come. We remember, right? I told you right now. The second male gamete fuses with the central cell's polar nuclei, right? The male gamete over here also has n number of chromosome. So this time it is n, and here you have 2n already. So n plus 2n becomes what? 3n. Becomes what? 3n. And what is it forming? The nucleus that is forming, it is known as the primary endosperm nucleus. The nucleus which is formed over here, it is known as the primary endosperm nucleus. The cell which will be formed over here will be primary endosperm cell. So the nucleus's name is primary endosperm nucleus and the cell's name is primary endosperm cell. So how much number of chromosome it has? 3n. Now over here, you have to have your attention. It is said the chromosome number in the gametes is 17. So that means haploid, if you see, it is 17. Okay, haploid, it is 17. Over here also it will be 17. 17 plus 17, what do you get? 34. 17 plus 17, 34, right? So here in the zygote, it will be 34. Now over here, you are asked about what is the chromosome number in its primary endosperm nucleus, this one. So 3n. So if n is 17, so 3n will be what? If n is 17, that is already given over here. So you have to do 3 into 17. That will be 51, not 61. It is 51, 5-1. Okay, and that is how you get your answer. So always understand the gametes will have half the number of chromosomes, that is n. So you will get the value of n. Okay, and always remember for PEN, it is triploid, 3n. So you have to multiply 3 into n. We are solving it for the first time. That is why I explained you in this depth. But you should understand it. Okay. So 3 into 17 is what? 51. So your answer would be A, 51. Got it? Yes. Okay. Next. If a tetraploid plant, what do you mean by tetraploid? 4n. Okay. Tetra, 4. Tetraploid plant contains 48 chromosomes in its nucellus. It's not nucleus, it is nucellus. It's a type of cell. Then the number of chromosomes in the egg cell and in a synergid respectively will be. That means I told you, if in a normal plant cell, if it is 24, then in the case of all the cells of the embryo sac, that is egg cells, synergids, antipodals, okay, all of it will be having half of it. That is 12. So over here you do not have to be worried that it is tetraploid or diploid or triploid, nothing. Over here it says that normal cell, okay, nucleus cell is nothing but a normal cell of a plant. It will be having the total number of chromosomes, that is 48. But egg cell and synergids, they will be always having half of it. So what's the half of 48, my dear children? What is the half of 48? It will be 24. Here also it will be 24. You do not have to think synergid have 2 in numbers, so 24 plus 24. They are separate. You are just asked about a synergid over here. So egg cell will have 24, synergid will also have 24. So your answer becomes B. Now to change the question, if at all it says that over here it was 34. They have not mentioned tetraploid and whatever is there, okay, they have mentioned it is 34. The number of chromosome is 34. What do? What is the number of chromosome in the egg cell and synergids? You make it into half. Okay, that will be 17 and 17. I hope you got the funda. If you are asked about the central cell, remember it has 2 nucleus. So in that case, 2 nucleus. In that case, what will be your answer? So again it will be 48. You have to tell me the answer in the comment section. That is something I am living for you guys. Cool. So I hope you got this particular thing. Fine. Now let's go. Okay. In a dithecous anther, each pollen sac contains 1000 mmC. What is that? Microspore mother cell or pollen mother cell. Okay. What is the total number of pollen grains produced by the anther? This is very easy. Again, you have the anther over here, my dear children. It is said that each pollen sac, that is the microsporangium, each of them has 1000 pollen mother cell or mmC, microspore mother cell, all of them. Okay, what is the total number of pollen grains produced by the anther? So, one of the pollen mother cell gives rise to 4. So, basically 1000 of them into 4, you get 4000. Right, here also you get 4000. Here also you get 4000. Here also you get 4000. So, my dear children, 4000 into 4, if you directly do it, 4000 into 4, your answer will be 16,000. This is the number of pollen grains produced by the anther. Got it? Easy, no? It's not very difficult. Yes? Okay, cool. Now, I think with that we are completing the round 2. Tell me, how was the round 2? Was it engaging? Do you think that these type of questions are something that you should be prepared for and it's a good practice for you guys? Yes? And now we come to theory based question. Now this will be comparatively easy, but my dear children, you have to pay attention to the details over here. Okay, so are we ready for question number 8? Let's see who have got 7 on 7 until now. I hope a lot of you guys have got 7 on 7. If at all you have not got, please make sure you're writing the question number, the wrong question number somewhere and re-attempting it later on. So, which pair of the following cells in the embryo sac are destined to change their ploidy after fertilization? That means they are going to change from haploid to diploid or diploid to triploid, something like that. Which of the cells are going to change their ploidy after fertilization, that is after fusion? So, you know, egg cell is something that will come together with the male gamete, N and N, it becomes 2N. So, egg cell is going to change its ploidy. I gave you one example. What is the next cell which is there which is going to change its ploidy? So, all of them were haploid except the central cell. Central cell was diploid in nature first, before fertilization. Now, after fertilization, my dear children, there are what cells that are going to change their ploidy? Egg cell and central cell, antipodals and synergids, synergids and egg cell, central cell and antipodals. Remember, antipodals and synergids are not involved in any fusion out there. So, whatever was their ploidy, it remains the same, haploid, haploid. So, what is that something that will change? Egg cell and the central cell. Egg cell was N, the male gamete came in, it became 2N, it formed a zygote. Central cell was already 2N because it had 2 nucleus, one more N came in with the male gamete and now it is triploid. Okay, now it is triploid over here. This is diploid and this is triploid. Someone out there asked me, can you make a list of cells out there which is haploid and triploid? I will be making it towards the end, do not worry, okay? So, that is there. So, I hope it is clear. So, these are the only cells that will be changing. The ones that are involved in fusion will be the ones that would be changing their ploidy. Okay? And it is going to form triploid what? PEN, primary endosperm nucleus or primary endosperm cell, either way. Going ahead with the next question. The ovule of angiosperm is technically known as the ovule which is there. Okay. of the angiosperm, the flowering plants, what is it known as? Megaspore mother cell, megasporangium, megasporophyle. It's very easy. What do you call it? In the case of male, you have the micro word. Okay, in the case of female, it is mega. Okay, females are mega powerful. Okay. So megasporangium is the answer, my dear children. Ovule is technically known as megasporangium, where everything is happening. So initially you have cells of nucellus. It is not nucleus, always remember while reading, it is nucellus. It is there for nourishment of what? Of the egg cell and everything later on. So over here there will be one cell that will be forming the embryo sac later on, giving rise to the female gamete, female gamete, synergids, antibodies and lot of things. So this whole structure, my dear children, the ovule itself, it is known as megasporangium. Okay, so your answer would be B. Not megaspore mother cell. The megaspore mother cell is right over here. This megaspore mother cell will undergo division and later on form the embryo sac in the further cases. Okay, remember that. Now going ahead, typical mature embryo sac of angiosperm is what? If you talk about the embryo sac, always remember this should be in your head by now. 3 antipodals, 1, 2, 3. 3 cells, okay. Here you have got 2 cells, synergids. There one more cell, egg cell. So 3 plus 3, 6 cell you have got, and over here you have got the last big cell, and that is the central cell. So 6 plus 1, 7. If you talk about the nuclei, 1, 2, 3, 4, 5, 6, and over here they have 2 nuclei, so 7 and 8. 8 nucleus condition is there and 7 cell condition is there. Always remember that. 7 cell and 1 extra nucleus is there. So 8 nucleated and 8 cell? No. 8 nucleated and 1 cell? No. 8 nucleated and 7 cell structure? Yes. So D becomes your answer for the 10th question out there. Okay, cool. There are 14 more questions, my dear children. We have to buckle up. Okay, going ahead. One of the 2000-year-old viable seed discovered during the archaeological excavation at King Herod's near the Dead Sea is. So I told you there are 2 seeds which was legendary. One stayed viable for 2000 years, the other stayed viable for 10,000 years. So here you are asked about the 2000-year-old guy out there which was found near the King Herod's place near the Dead Sea, my dear children. What was it? Was it maize, sunflower? No. Was it Lupinus articus or was it Phoenix dactylifera? It was Phoenix dactylifera. So children usually have confusion between this and this. So Lupinus articus is the one that stayed viable for 10,000 years. It is found near the Arctic region, in the colder region, Arctic tundra region. This is Phoenix dactylifera. It is also known as the date palm. In my videos, in the theory videos, I have given you the pictures as well so that it becomes very easy for you to understand. This is a very beautiful flower Lupinus articus have. And how do you remember it, that this is 10,000 and this is 2,000 always? So if you say A, B, C, D, my dear children, L comes before P, right? A, B, C, D, E, F, G, H, I, J, K, L, M, N, O, P. L, M, N, O, P, right? L comes before first. So first position goes to Lupinus always with 10,000. It remained viable for 10,000 years. Phoenix, my dear children, it remained viable for 2,000 years. So here you are asked about Phoenix. So do not forget it. Going ahead. Identify the correct order of events in pollen-pistil interaction from the options given below. So, my dear children, okay. So, pollen-pistil interaction, I hope you remember. There is pollen, okay. In the pollen, what cells do you have? You have vegetative cell, okay, and then you have generative cell. This generative cell divides into 2. You have one male gamete, you have the second male gamete. The pollen grain which is there, it will go land on the stigma. It lands on the stigma. And now what happens, my dear children? Remember what is there in the pollen grain? It is surrounded by 2 layers, okay. There is a continuous layer which is known as intine and there is a discontinuous layer which is known as exine. Now in this discontinuous layer you see pores. From this pore, my dear children, this vegetative cell will slowly come out like this. And later it will be carrying these 2 male gametes over here. So slowly this particular thing, vegetative cell which is there, it is also known as tube cell. What do you call it? You call it tube cell. Why tube cell? Because this is the one that will be helping in the formation of the pollen tube. So this cell will slowly form the tube, and over here who would be coming along? It will be the male gametes. So pollen tube comes in, it goes into the style, and then goes where? It goes to the ovule, okay, which is there in the ovary. It goes to this particular region which is there. There is the ovule. Over here it gets in the ovule. Inside the ovule, what is there? Inside the ovule, you have the embryo sac. So the next step is it gets inside the embryo sac and then it will be fusing. Okay, understand that. So identify the correct order of events in the pollen pistil interaction. Release of male gametes into the embryo sac, this comes very late. Deposition of pollen grains on the stigma, that has to be first, right? Entry of pollen tube, no. Development of pollen tube, no. Entry of pollen tube into the ovule, no. So over here, remember, first thing, you have to deposit the pollen grain over here on the stigma. So this would be the first thing. Second one is the first thing. So you can eliminate these two options, because over here, second one is not the first thing. You have now C and D. Okay, now the next step after this is you can check the options and see. Here it is 4, here is 4. So deposition of pollen grains and then development of pollen tube. Is that correct? Yes, that is correct. Now if you go with fifth one over here, after development of pollen tube, it says that entry of pollen tube into the ovule. Okay, and here what does it say? It says that entry of pollen tube into the embryo sac. So that is wrong. First it has to enter the ovule and then the embryo sac, right? So this particular statement again has become wrong. What is the correct one? This would be a correct one. Okay, so fifth one, that is entry of pollen tube, where is it? Into the ovule first, and then what happens, my dear children? Entry of pollen tube into the embryo sac, and then what happens? Once it is entered, the release of male gametes into the embryo sac, there the fusion will be happening. So your answer would be C. So sometimes the option itself can give you answers. If at all, you know it well. Usually children get confused with ovule and embryo sac. Do not have that confusion. You have the ovule, the megasporangium. Inside that you have the embryo sac with the female gamete and synergids and the antipodals and everything out there. Cool? Yes. Now, okay. How many microsporangia are located at the corners of a typical bilobed anther of angiosperm? So my dear children, over here, please tell the answer is 4. Why? Because most of you get confused with 2. It's just not at all 2. You are asked about over here, or some of them give answer as 1, because in one corner only one microsporangium is there. No, you are asked about a bilobed anther. So in each corner you have 1, 1, 1. So in total your answer would be 4. Okay, how many microsporangia are located at the corners is the question of a typical bilobed anther of a angiosperm. Your answer would be 4. Okay, cool. Next question. Identify the correct statement. Are we ready? Yes. Only one megaspore present towards chalazal end remains functional. What do you mean by that? So for that you need to have an understanding of chalazal end and everything. So my dear children, this is the style. You have the ovary over here. You have the ovule over here. Right? Always remember, the ovule is somewhere where everything is happening, right? Where the baby is going to form and everything is going to happen. So over here, it has to provide nourishment. So there is a structure that is known as placenta. Placenta provides nourishment to the ovule through a structure that is known as funicle. We have studied this in the theory session, right? The funicle attaches at a point which is known as hilum. Okay? The ovule says, I am not protected, I want two coverings. So you have inner integument first, and then you have an outer integument. Okay, there is an opening over here. You might have seen that opening, and this is known as micropyle. And this region where you have the opening, it is known as the micropylar region. What do you call it? Micropylar region. The opposite end of the micropylar region is known as, this region which is there, it will be known as the chalazal region. Which is why I taught you everything for solving this particular question. Now, if this theory is not clear, will you be able to answer this? Okay, now that is chalazal end. Now, over here we have to get inside the ovule. In the ovule, my dear children, you would be having the cells of the nucellus. The cells of the nucellus. One such cell, okay, there will be one cell, only one cell, that is known as the megaspore mother cell. It will undergo division. Megaspore mother cell is also denoted by MMC. It will undergo meiotic division. You get first 2 cells, then the next round of meiosis, you get 4 cells. You get 4 cells altogether. Now in these 4 cells, 3 of them degenerate, only 1 remains. Only 1 would be remaining. Okay, and where is it all happening? towards the micropolar region. Always remember, all the important things happens towards the micropolar region. So basically the MMC divides, it forms 4 cells, 3 of them degenerates, 1 cell remains active, that is known as what? Functional megaspore. It is known as functional megaspore. This functional megaspore is going to form the embryo sac in total. All of it happens towards the micropolar region. Okay. So at the end, when the embryo sac is formed, you will be having the synergids over here, the egg cell over here, the antipodals over here, and the polar nuclei over here. So always remember, the opening wherever it is there, the micropolar region where it is there, that is where you have the egg cell. Okay, because it has to fuse with the male gamete, right? So that is where you would be having the egg cell. Antipodals are towards the chalazal end. So picture can be like this or like this. Please understand, wherever you have the egg cell, that is where you have the micropolar region. So only one megaspore present towards chalazal end remains functional. Wrong. It is not towards chalazal end, it is micropolar end. Now 3 megaspore present towards the chalazal end degenerate gradually. Yes. So 3 of them which are present towards the chalazal end, they will be degenerating, but 1 of them, which is a functional, my dear children, that will be remaining. That will be remaining, and that is towards the micropolar region. Now each megaspore mother cell directly develops into a megaspore. Each megaspore mother cell, do you think it directly develops into megaspore? No, it has to undergo division, right? This is wrong. Each female gametophyte is 7-celled and 7-nucleated structure. No, I told you it's 7-celled but 8-nucleated structure. So what is correct, my dear children? Only B. Only B. So 3 megaspore present towards the chalazal end will degenerate. All the degeneration, all the sad things will happen towards chalazal end. Okay, what remains functional is over here in the micropolar region, and that functional one will give rise to what? Embryo sac. Always remember that. So answer is B. Cool. Next. Which of the following aquatic plant does not show pollination by water? So in your book there are certain examples of plant which shows pollination by wind. In wind it is grasses, okay, corn cob, okay, they are all examples of wind pollination. Now in plant, in water pollination, you have Vallisnera, you have Zostera, you have Hydrilla. What about water hyacinth? Even though the term water is given, we believe that water is given, so this will be answer. But this is something that does not show water pollination. It usually shows insect pollination or wind pollination, but not water pollination out there. So this is wrong. So which of the following aquatic does not show pollination by water? It is water hyacinth. So Vallisneria and Hydrilla, they remain at the top of the water. The pollen grains come and land on the top of the water. It will slowly move with the water to the female flower. In Zostera, which is also known as sea grass, it happens in the submerged condition. Inside the water surface only the pollination would be happening. But all of them, all the other ones, they are showing water pollination, except water hyacinth, water lily also. Okay, always remember that. Now my dear children, with that I remember, I have made you solve certain questions in the theory sessions as well. It is very much important for you to practice them also. Okay, so please watch the theory if you have not watched. Now identify the mismatch. So from here I can teach you the haploid diploid thing. Someone was asking me about the haploid diploid, I can teach you here itself. So my dear children, identify the mismatch. PEN, primary endosperm nucleus, it is triploid, is it? Yes. How is it triploid? Because it is formed of the 2 polar nuclei, which is diploid in nature, and then it is made of 1 male gamete, which is haploid in nature. So haploid and diploid comes together, you form triploid primary endosperm nucleus or primary endosperm cell. Fine. Next, antipodals haploid. Are the antipodals haploid? Yes. The antipodal cells which are there over here, each of them would be haploid. So this is also correct. Okay. Now going ahead, you have zygote which is diploid. Is the zygote diploid? Let's see. Zygote is formed of the male gamete. Male gamete has haploid. Okay. It has haploid number of chromosomes, and it is formed of female gamete. Female gamete, again it has haploid number of chromosomes, so N plus N you get diploid, 2N number of chromosomes. Now synergids. Synergid, my dear children, again all of them, they are haploid in nature, but over here it is mentioned as diploid. So this would be wrong, because this should be what? Haploid. So this is the mismatch that we have in D. So remember, antipodals, okay, I'll write it somewhere if you want. Antipodals haploid, okay, and then what we have, egg cell haploid, and synergids haploid. What is not haploid in the embryo sac? There is only one exclusion, and that is the central cell. The central cell, it is diploid, but if you talk within the central cell about the polar nuclei, both of them are haploid in nature. So two haploid, so two polar nuclei will be there, so that is why haploid plus haploid becomes diploid over here in the case of central cell. Okay, now if you want to know about the male gamete, that is about the female part, right? Now if you want the male part, the male gametes, obviously, male gamete, it is what? Haploid in nature. That's all. Okay, if you want to know the ploidy of any more other cells out there, definitely let me know in the comment section. I'll be helpful for you. Okay, next, identify the correct order of steps involved in artificial hybridization in plants. What do you mean by artificial hybridization? Where you are basically taking the pollen grains and putting it out there, okay, and you are doing artificially. It's not the wind, it's not the water, it's not any other organism that is doing the pollination. You are doing it because you think that this plant has great varieties. If both of them come together, you can get a good hybrid variety of plant. Okay, so you have to tell the correct steps involved. So artificial pollination first, then emasculation. What do you mean by emasculation? Masculation, you know, masculine feature. Emasculation means you are removing that masculine feature. That means you are removing the anthers, basically you are removing the stamen. Removal of stamen is known as emasculation. So you only tell what will be the use of it. Artificial pollination, you are pollinating it. Okay, and then basically removing the stamens and then rebagging, and bagging does not make any sense, right? So first thing that you have to do, if you are worried that this is a flower over here and it has the female and the male part, and if you are worried the pollen grains from here can land over here, the first thing that you will be doing is what? You will be basically chopping this particular portion off, the male parts off, and that is known as emasculation. So that will be the first step that you will do. So over here there's only one point that gives emasculation as the first step. So we will say emasculation, this is correct. Okay, and after that what do you do, my dear children? Such that no other pollen grains comes and lands over here. We have removed this thing and now we do not want any other pollen grains to come and land over here. Okay, we are giving the stigma the time to become receptive. So we will bag it. This is known as bagging. So emasculation, then bagging. After bagging, what we'll do when we get to know the stigma is finally receptive, it's grown up. Okay, what we'll do, we will slowly remove this bag. We will take the pollen grains that we want and we will do artificial pollination. We will put it. So this is known as artificial pollination. And after that, we do not want any other pollen grains to come again. So what do? We again cover it. This is known as rebagging, so that no pollination can happen later on. So slowly this will form the pollen tube and the whole fertilization would be happening. So your answer would be C, not A, not B, not D, and I hope you know why. Because first step always has to be emasculation. Okay, if it's a bisexual flower means if it has two anthers out there. Bisexual flower means if it has both the male and the female parts out there. So over here, suppose it is unisexual flower, it does not have the male part. In that case, you do not have to do emasculation. Okay, if it is bisexual, if it has two of them, two male and female, it will be known as bisexual. Okay, two sexes. Okay, next. Among the following related to pollens, choose the correct one. Okay, so statement 1 states, okay, this is statement 1 and this is statement 2. Okay, statement 1 says, in 40% of angiosperms, pollen grains are shed at 3-cell stage. Is it? What do you mean by 3-cell stage? So, the pollen grain which is there, I told you there is vegetative cell and there is generative cell. In some cases, my dear children, they are shed like this. They are shed like this. There is only one cell and second cell. When they enter the hysis, the pollen grains goes off and it just has 2 cells out there. This is in the case of 60% of the flowering plants. 60% of the flowering plants goes like this. In rest 40%, my dear children, what happens? This will divide into 2. So now you have 1, 2 and 3 cells condition. So is this statement correct? In 40% of angiosperms, pollen grains are shed at 3 cell stage. Correct. Now, intine is made up of cellulose and pectin and it is discontinuous with germ pores. Now that is the next question. So I have to remove a thing over here. Let's see. Okay. Ayo, wait. So this is your pollen grain. A pollen grain has the vegetative cell, the generative cell. It has its own plasma membrane also, please remember that. Okay, PM, plasma membrane. And then it has a continuous layer, that means it is not broken anywhere. This layer is known as intine. And this intine, my dear children, is made up of 2 things. One is cellulose and the other one is pectin. Okay, CP, cellulose and pectin. Now you have one more layer, very important layer out there, which is not continuous, it is discontinuous. What do you call this layer as? You call this as exine, the outer layer. And it is made up of the toughest material on earth, which is known as sporopollenin. You know what? Sporopollenin can resist alkali, acids, anything, which is why the pollen grains remain viable for so long, because they have sporopollenin within them. Okay? And now, wherever you do not have sporopollenin, that's where you have a gap. Okay, and this gap which is there, it is very important. It is known as germ pore. It is known as germ pore. Whenever the pollen grain goes and lands somewhere, the vegetative cell which is out there, my dear children, it will be forming a tube form of structure. And this tube will be moving out through the, what do you call, germ pore. So this is very, very important. So intine is made up of cellulose and pectin and it is discontinuous with germ pores. That will be a wrong statement. Why? Because it is not the intine, it is the exine which is discontinuous. The outer layer which is discontinuous. Okay, always remember that. So in the mall and all, if you see, inside you would be having the doors, we do not have much checking out there, but in the outer layer you would be having checking doors and things like that, right? Because you have to have tighter security. Similarly over here, in the case of exine, they would be having more doors there. In the inside, it's all cool. I do not know if it makes sense, but yeah. Okay, you can have different understanding of it as well. So, so that is the question. Statement 1 is correct and statement 2 is what? Incorrect, my dearest little champions. And now, question number 19th. Which of the following characters is not required in autogamy? What do you mean by autogamy? Where you have both the female and the male parts and the pollen grains are landing up over here in the same flower, that is known as autogamy. So you are asked which is not required in autogamy. What is the feature that is not required? So flowers require synchrony in pollen release and stigma maturation. Arey bhai, it's required. This pollen grains have to land there and the stigma should be receptive, then only fertilization will have to happen. So this is a feature that we need. Anthers and stigma should lie close to each other. Obviously, if they are not lying close to each other, do you think autogamy can happen? So yes, this is also a feature that they need. Flowers should be bisexual. Obviously, they should have both the male and female, then only autogamy can happen. Requires pollination agents. Do you think they require pollination agents? No, they lie in close proximity that sometimes they do not even require pollination agents, my dear children. It can happen like that also. Okay. If pollination agents are there, that is totally fine, okay, but they definitely, it's not a very important thing for them to require a pollination agent because they lie very close to each other, okay. So this is something that would be out. So this is not required in the case of autogamy. Something that is partially correct, partially incorrect, but okay. Compared to other options, when you have something like that, compare it with the other option. If you think that this is the option that stands a bit out, go for that. Now, question number 20. Which one of the following statement is correct? Chasmogamous flowers always exhibit gytonogamy. What do you mean by chasmogamous flowers? First of all, you should know that. Again, so my dear children, there are flowers which are open, they are blooming, okay. Suppose they have both the male and the female part, let's put it like that. And there are flowers which would be closed, but they still have the female and the male parts out there. Okay, what do you call these closed flowers? You call them clistogamous. Cleistogamos. Cleistogamos. So Cl is there. Cl stands for closed also. Always remember Cleisto are the closed ones. Okay, and here they are open ones. So Jasmogamous flowers, my dear children, from here to here pollination can happen. Agreed? Autogamy can happen. Now suppose there is another flower over here and it has the female and the male part. From here to here also pollination can happen. That means gytonogamy is also happening. Okay, two flowers of the same plant, gytonogamy. Now there is another plant with another flower out there. From here to here also pollination can happen. That means xenogamy can also take place. Two different plants, okay, from there pollination can happen. So in Jasmogamous flower, three things are possible. Autogamy only if you have both the male and the female parts together, okay. Autogamy can happen, gytonogamy can happen, and also xenogamy can happen. Okay, so Jasmogamous flowers always exhibit gytonogamy would be a wrong statement. It can exhibit other things also. Okay, it can exhibit other things also. Now Cleistogamous flowers always exhibit autogamy. What about the closed ones? We forgot about the poor ones, right? Cleistogamous, do you think gytonogamy can happen from here to here, things can come in? Or from there, from the other plant it can come in? No. Here they are always closed, my dear children. So they have to exhibit, always exhibit something that is known as autogamy. So this statement would be correct. Cleistogamous flowers always exhibit autogamy. Jasmogamous flowers never exhibit autogamy. They can, if they have both the sexes out there. Cleistogamous flowers exhibit both autogamy and gytonogamy. No, Baba, it's closed. And always remember one more point, okay? Cleistogamous flowers are mostly within the roots, okay? You will find them towards the root region, that is within the soil, or towards this particular region, just above the soil. So rhizomes or the root is where you find the Cleistogamous flowers, because anyway they don't need a pollinating agent, so they can be inside. And towards the aerial branches, aerial side as in the one that is above the soil level, there you will be finding the Jasmogamous flowers. This is a question that I solved in the theory part, that's why I remember it. Okay, so your answer would be B. Have a look at the picture given in your NCERT about the Jasmogamous flowers and also examples, Viola, Oxalis and Commelina. You feel you're Spanish when you talk about them, but yeah. Now my dear children, we just have 3 more questions apart from this. So are we ready? Yes. So in castor and maize plant, this is a tricky question. If you are someone who just have read through NCERT in this level, in the upper level, if you have not gone in depth, it will be difficult for you, I'm telling you. So in castor and maize, male and the female flowers are born on different plants. Autogamy is prevented but not gytonogamy. The anthers and the stigma are placed at different positions to encourage cross pollination. Both autogamy and gytonogamy are prevented. What do you know about castor and maize plant? So castor and maize plant, my dear children, you call them monoecious plant. Monoecious plant. So I told, if children are there who have read NCERT like this, they won't be able to answer. But I'm pretty sure every one of you have given your best in reading NCERT, have given your best in listening to the lectures, and you know the answers as well. So my dear children, they are monoecious in nature, as in their plant, if we talk about their plant, there will be a flower that will be that will be female. There will be a flower that can be male. So you have both the sexes. You have both the sexes in one particular plant. This condition is monoecious. Okay, this is where you have unisexual flower. Unisexual flower in monoecious plant. In a monoecious plant. And there is one more type of monoecious, my dear children, where what is happening? There is plant, there is flower. You have both the sexes in one particular flower itself. In that case, you call them, anyways here also you have both the sexes in one particular plant. Here also you have both the sexes in one particular plant. So they are also monoecious. They are also monoecious, but here you have bisexual flowers. Here you have bisexual flowers. Now castor and maize comes in this category. Unisexual flowers in monoecious plant. In this case, the male and the female flowers are born on different plants? No, they are on the same plant, right? Autogamy is prevented but not gytonogamy. Is autogamy prevented, my dear children? Yes. Why? Because this flower do not have both the sexes. For autogamy to happen, you need to have both the sexes. Do they have it? Does this have it? No. So autogamy is definitely prevented, but gytonogamy, is it prevented? No. From here to here still it can go. You have both the sexes, right? So I can say, yes, autogamy is prevented but not gytonogamy. This is a correct statement. Now going ahead, the anthers and the stigma are placed at different position to encourage cross-pollination. Yes, the anther and the stigma are positioned in different positions so that they have cross-pollination there. Done. Now, at least gytonogamy as I told, it's a form of cross-pollination but not entirely correct, but yeah. Both autogamy and gytonogamy are prevented, that is not possible, right? So my dear children, always remember, okay, this is known as, what do you call this? Outbreeding devices. This is a form of outbreeding devices. Plants, basically they were like this, but they have evolved into something like this, so that newer chances can be given. If at all pollination happens like this only, you will have the same genetic feature, and this can lead to inbreeding depression, which is why plants are that they can do this. Now over here, from other plants also, my dear children, pollination can happen, xenogamy can happen, and also gytonogamy can happen. So always remember, castor and maize, they are monoecious plants with unisexual flowers. So if at all you do not know the concept, you would not be able to answer, but now since you know the concept, you are all ready, right? Children, I hope you are understanding. I am going in depth to explain you guys. It might be a time-taking process, but it's for you guys, okay? Now the germ pores in the pollen grains are the regions, you have to tell me the answer, which are made of lignin and suberin. They are saying the germ pores which are there in the pollen grains, it is there in the exine region, no? It is the region which is made up of lignin and suberin, or that can withstand high temperature and strong acids and alkali. Do you think the germ pore is a region that can withstand? Anyways, which lacks sporopollen, through which sperms are released into the female gametophyte. What is your answer? Your answer would be the one that lacks sporopollen. The germ pores are the region where you do not have sporopollen. Elsewhere you all have the sporopollen things. That is why you have a exine out there. So this will be your answer. My voice is cracking. Now we are going to go ahead with the last round, that is diagram based question. My dear children, you have 2 questions out there and you have to solve it. Okay, if you were with me towards the last session, you would be able to answer this. I am pretty sure about that. Okay, now if at all you're not, it's totally fine. We are gonna learn it today. Are we ready? I hope we are ready. And my dear children, I hope you had fun solving all the questions out there. Do not forget to hit the subscribe button. I'm reminding you again. Okay, and also do not forget to hit the like button. Now, if at all you have doubt, put it down, okay? Now, in this diagram showing the LS, okay, of an embryo of grass. Grass is what? Monocot. Always remember. Identify the answer having the correct combination of alphabets with the right part. Okay. So, what's the easiest part to identify? That is something that we should be identifying first. Always remember, you're talking about a monocot embryo. In monocot embryo, I have taught you. You just have few things. Okay, what are those things? So, over here, you will be having an embryonal axis. This region is where you will form the plumule. Plumule is the future shoot. This is the region which is known as radicle. Radicle is the future root. Radicle is always covered by something that is known as root cap. Okay, and monocotyledon means it just has one cotyledon. It does not have 2 cotyledon, 1 cotyledon. And this one big cotyledon which is there, it is known as scutellum. What do you call it? Scutellum. It has given that name. Okay, it has just only 1 cotyledon, that is why it has given a name, that is scutellum. Okay, the other cotyledon also tried to come out, but it was not able to. And that rudimentary cotyledon which was not able to make it, it is known as epiblast. What do you call it? It got blasted off. Okay, it was not able to make it. Epiblast. Now, if it is just having 1 cotyledon, what is that something that provides nourishment? It is the endosperm as well. So, it has the endosperm still left. Okay, and the cotyledon also there. This cotyledon is known as scutellum. Now, around the plumule, you will be having leaf primordia, something that gives rise to the leaf, and also something that is known as coleoptile. Coleoptile. Over here, near after the root cap, you will be having something that is known as coleorhiza. Always remember R for root and P for plumule. So, coleoptile is towards plumule region and coleorhiza is towards the root region. So, this is the general structure of the monocot embryo. Okay. Now, in this picture, over here, this D part that you see, it is the scutellum, the only cotyledon that it has. So D is scutellum. Where do you find D as scutellum? You have to find that. So here D is radicle, here D is coleorhiza, here D is scutellum, and here also D is scutellum. So I can exclude these two options out there. Now the next thing that I am pretty sure is A. What is A? A is the rudimentary, okay, rudimentary cotyledon out there, the one that was not able to make it. So it is epiblast. So A is epiblast. Let's see that. A is epiblast. You have only one option, and with that I found the actual answer without even going ahead with anything else mentioned up over there. Okay, so here A is there is two A, please remember. A is shoot apex. Anyway, this cannot be shoot apex, it is root apex. So again, that is also wrong. So going ahead, A is epiblast, okay. B that you see is the radical region. C that you see is the coleoptile. So here you will be having the plumule, right. The D region that is there, it is to the scutellum, and what do you call E region which is there. So E, they have not mentioned it. Just a second. This is E. Okay, so E region which is there, it is coleorhiza towards the root apex, yeah, and F also not mentioned, but F is what? The shoot apex. So F would be this line that you see, it is F. Okay, so your answer would be C. Without even going ahead with any more option, with the help of D and A, you got the answer. So that is why I am telling you, if at all you understand the picture well, if you understand the theory behind the picture well, you would be able to answer everything out there. Now with that, I have explained you over here. Monocot has one cotyledon, then you have endosperm, and then you have radical, plumule, coleoptile there, coleorhiza here, root cap here. You will be able to solve one more question. Are you ready for that? Let's see. So for this, the answer is C, and for the last question that we have today is, in the given diagram, identify the parts labeled as A, B, C, D. So first of all, is it a monocot embryo or a dicot embryo? It will be a monocot embryo, because you see only one side you have a cotyledon. This part is known as cotyledon. If you had it on both the sides, it will be dicot. Okay, it will be dicot. Okay, so this is a monocot embryo. B thing that you see is again the scutellum, and apart from scutellum, what do you have? You have a whole big endosperm out there. So this whole region is endosperm, but here they have not actually mentioned C for showing endosperm. C is the outer layer. That is what C is. So if you zoom in the picture, you will get to understand that. There is the picture. Okay, C is just the outer part. B is the region over here. B is what? Scutellum. Now A is what, my dear children, and D is what? So A will be something related to the what do you call, the shoot region, and D will be something that is related to the root region. We have got that much idea, and we can find the answer even without knowing what is C. Let's see. So over here B is scutellum. B is scutellum. I have two options like that. So I will be cutting off these two options. Are you? Yeah. Okay. Now I have got A. A should be something related to the shoot region. So A over here is coleoptile. A over here is also coleoptile. So I'll say okay, I have to keep that option. Let's go with D. What is D? D over here should be something that is related to the root. Okay, so D is coleorhiza. Over here D is pericarp. We do not know what's pericarp, ma'am. It's okay, you know what's coleorhiza, you got the answer. This will be your answer. Okay, coleorhiza is after the radical region that you have. So here you have coleoptile, here you have coleorhiza. This is the scutellum. What is this? This is the endosperm. But what is C? You should be knowing what is C. As of now you didn't know, that's fine, but you should be knowing it, right? What is C, my dear children? So this whole thing that you see, it's the ovule. It's the ovule, right? And the ovule which is there, it is inside what? The ovary. Okay, the ovule which is there, it is inside the ovary. So ovule will be having its own covering that is known as testa and tegmen. But apart from that, the ovary which is there, it also has its own covering. It is known as pericarp. It is known as pericarp. So C is nothing but the pericarp region which is there. Okay, always remember, you have the ovary, then the ovule. So ovule will be having its integuments which will later turn into testa and tegmen in the case of a seed. What is a seed? When the ovule undergoes fertilization, when the egg cell and everything fusion happens, there will be zygote formed, right? So now you won't call it as an ovule, you will call this as a seed. So seed will have its own covering, and then the rest of the covering which is there together, the whole covering of apart from the ovule, it will be known as pericarp. So pericarp is nothing but the wall of ovary. It is again bifurcated into different parts: epicarp, endocarp, mesocarp, and things like that, about which I taught you in the theory session. I want you to go have a view on it because it will be really important for you to get marks. Okay, and with that we complete all the 24 questions out there. Yes, and now scoreboard. You have to tell me what did you score out of, or how many marks did you score out of 24. Put it down in the comment section. That is how I'll get to know. And also, if you have any doubts still lingering around the topics, please let me know in the comment section. I'll be really happy to help you out. Okay, and my dear children, thank you for playing this game with me. Until next time, we'll meet again soon. The next chapter, which is human reproduction, we'll go in line by line, so that in the school also I hope this trend is followed. If in your school some other chapter is being followed, definitely let me know in the comment section. And if you're someone who's preparing for KCET, what do you have to do? You have to subscribe to our channel, that is Diksha Karnataka. Wishing you all the best. Take care of yourselves. Bye bye. Lots of love.